Hello dear friends, welcome back to my channel NB Biology Tutorial. I am Professor Namrata Borache. Today we are going to continue the next topic, that's the origin and evolution. In this topic we see the phenomena that's a modern synthetic theory of evolution. So let's begin. As this modern synthetic theory, it is the result of the true synthesis of all biological disciplines studied pertaining to genetical, ecological, anatomical, geographical, paleontological. They pursue to explain the mechanism of evolution. It is important. All these are important. This importance was given to both mutation and the natural selection. Our feature JBS Heldon, T. Debowski, J. Huxley, E. Mayer, Simpson, Stebbins, Fisher, Sewall Wright, Medill, T. H. Morgan are the main contributor of modern synthetic theory. But in this, Stebbins, in his book That's a Process of Organic Evolution, discussed 5 key factors such as the gene mutation, second one is mutation in the chromosome structure and number, third one is genetic recombination, next one is natural selection, and the last one is reproductive isolation. All these are contribute in the evolution of the new species. All individual of some species constitute a population. The population occurs in a small group of interbreeding population. That is a small group of interbreeding population. It says as the Mendelian population. Total genetic information encoded in sum total of gene in Mendelian population, it says as the gene pool. It is also defined as the total number of gene of all individual in a population. As the gamete produced by individual furnish a pool of gene of next generation from which the chain will be selected. The gene pool is changed. In the two cases, first one is a replacement of one generation by the other in Mendelian population, and the second one is a migration of population. Effectively alter the gene, both these two, it is alter the gene pool. The next one is the gene frequency. The gene frequency is says as the proportion of an allele in gene pool to the total number of allele at a given locus. Is called as the gene frequency. As a modern synthetic theory, it comprises the 5 main factors. These 5 main factors is the first one is a gene mutation, second one is a mutation in the chromosome structure and the number, third one is genetic recombination, fourth one is natural selection, and the fifth one is reproductive isolation. As these 5 main factors, which is broadly divided it into the 3 main concepts. The first concept is the genetic variation, second one is natural selection, and the third one is isolation. In this video, we concentrate only the first point, that's a genetic variation. This genetic variation, it is caused due to various aspects of mutation, recombination, and the migration. So, let's begin. The first phenomena, that's a genetic variation. What's a genetic variation? It's a change in the gene and the gene frequency. These are small segments of the DNA. If it is changed, that is a change in the gene. And the gene frequency, whenever it's changed, there is a genetic variations. It's occur. This genetic variation, it is occur due to the 5 factors which is responsible for the genetic variation. First one is a gene mutation, second one is genetic recombination, third one is gene flow, next one is a genetic drift, and the fifth one is a chromosomal aberration. So, the first phenomena, it's a gene mutation. As a gene mutation means a permanent, sudden, then the sudden permanent heritable change is called as the mutation. It is a spontaneous permanent heritable change. It called as a mutation. The mutation can occur in the gene, in the chromosome, and in the chromosome number. Mutation that occurs within a single gene. Single gene means what? That is said as a single base pair. Single base pair of a DNA, it called as the point mutation, or it called as the gene mutation. You can see here there is a, in the normal DNA, there is a presence of nitrogen base is the GAG. But if the mutation it is occurred in a single nitrogen base pair, that is instead of the T, there is a phenomena with the nitrogen base which is observed, that is the T. If this T it is paired with the adenine, that is a thymine, it's paired with the adenine due to the change, that is a mutation which is occurred in a single base pair. That's called as the gene mutation, or it says as the point mutation. The mutation which is occurred more than a single gene, that is says as the cross mutation. This leads to the change in the phenotype of the organism. Result is the variation. As this variation which is observed in case of example is the sickle cell anemia, means whenever there is a change the single nitrogen base pair, there is a change in the amino acid, and if the amino acid is changed, there is a change the normal RBC cell it into the sickle cell RBC cell, that is said as the sickle cell anemia. Afterward, the second phenomena we see, that is the genetic recombination. This genetic recombination in the sexually reproducing organism, during the gamete formation, there is an exchange of genetic material. It's here. There is the exchange of genetic material occurs between the non-sister chromatid of a homologous chromosome. So these chromosomes are the homologous chromosome. It is called as the crossing over. The point it says as the crossing over. It produces the new genetic combination. It produced new genetic combination. Result is what? There is a variation. The fertilization between the opposite mating gametes leads to the various recombination, resulting into the phenotypic variations. Result it into the phenotypic variation, causing the change in the frequencies of alleles. The next phenomena we see, that is a gene flow. This gene flow, it is a movement of a gene into, means inside the population or out of the population. The gene movement may be in the form of migration. Migration of organism means this organism, it is inside, it is goes inside, or it is come out from the population, or it's a gamete, or the gamete means a dispersal of the pollen grain or the segments of the DNA. That's a transformation. Gene flow also alter the gene frequency. Result is evolutionary changes. Next one is a genetic drift. This genetic drift, any random fluctuation, that is the alteration in the allele frequency occurring in the natural population. So by the pure chance, it's called as the genetic drift. For example, when the small size of population, it is severely reduced. When there is the small size population, this is the small size population, when it is severely reduced due to the natural disaster like the earthquake, flood, fires, cause the elimination of the particular alleles from the population, or the smaller population have the greater chance of the genetic drift. It will result in the change in the gene frequencies. Genetic drift is also important for the evolutionary changes. You can see here their predator. This is the predator. Predator eat more easily seen beetles. More easily seen beetles. Which beetles is more easily seen? That is the green colored beetles. That is the green color beetles means there is absence of the brown gene in that green beetle. Resulting is there is increasing the brown gene frequency. It's increasing, but the green color gene frequency it goes on decreasing. That is as the genetic drift. The next one is the chromosomal aberration. This chromosomal aberration means the structural and the morphological change in the chromosome due to rearrangement is called as the chromosomal aberration. This chromosomal aberration, it is occurs due to either the deletion, duplication, inversion or the translocation. The first phenomena is a deletion. You can see here. On the chromosome, there is a presence of the 6 gene A, B, C, centromere and D, E, F. As there is a loss of one of the gene from the chromosome, that is said as the deletion. The second one is the duplication. In the duplication, one of the gene, it is duplicate, it is repeated. It's double in number, that is a B gene. It is a double in number, so it says as a duplication. Third one is a inversion. What's the inversion? As there is a presence of 6 gene A, B and the C, and on the another side there is a presence of the G, E and the F. In the inversion, a particular segment of chromosome is broken. There is a broken the particular segment of the chromosome. Particular segment of chromosome is broken and get reattached to the same chromosome in an inverted position. This word is very important. In an inverted position due to 180-degree twist. There is no loss or the gain of a gene of the chromosome. The next phenomena we see, that is a translocation. This translocation, it's a transfer of a part of chromosome. There is a transfer, the part of chromosome. Part means this half part, okay? That is a T, E and the F. There is a transfer, a part of chromosome or a set of a gene. Set of gene means these are the 3 genes, okay? And on the another chromosomes there is a presence of the 3 genes. That is a set of a gene J, K and the L. There is a transfer of a part of chromosome or a set of gene to a non-homologous chromosome. It called as the translocation. Means here in the original chromosome A, B, C on the one side and on another side there is a presence of the D, E, F. But when there is a translocation phenomena, it's occur, instead of present the D, E, F, the chromosome or the genes will be J, K and the L. That is, it's affected naturally by the transposons present in the cell. Hope so you understood it. Thanks for watching my video. Stay tuned to my channel. Thank you.